Hi, welcome to the second video. So in the previous video I was explaining you about the basic requirements, what we need and how to develop the plugin and all. So as I told, I'm going to explain what's our major requirement, what's our algorithm needs to be developed, how it needs to be developed, what's the feature it needs to contain, the main basic feature. Okay, so as I have shown you, this is the Celtivan software, and these are all the reactions which are involved in that pathway, to like supersignaling pathway, and it contains different reactions, molecules and reaction IDs, everything you will get. Okay, so as I told in my previous video, we need to assign give values to each of these molecules based on the reaction. If the reaction is an incoming one, the molecule should be given a +1 value. If it is an outgoing reaction like this one, the molecule should be given a -1. So, and at the end, what's the total value of that molecule? So it should be -1 plus 1, that will be a 0. So like that we need to assign. Before that, so we need to, for this kind of reactions, what we were doing is, so we were manually calculating all those values for each of the molecules. So there is one pathway to like TGF beta signaling pathway we had. So there we were assigned. Okay, before that let me show you okay, yeah, this one. So what we were doing is, just for displaying purpose, simplicity, so each of the pathway we were simplified in this format as shown in the figure B. So each of those molecules are represented in alphabetic characters A, B, C, D, like that, and each of the reactions are named V1, V2, V1, V2, like that. It's been named just for understanding purpose. So one, so I will share this PDF file with you. This is one white paper research article was prepared. This is in communication. For this is the basic algorithm. So that we are going to develop now. So yeah. So this is the way we are representing B, A, B, C, D, and B1, B2, B3. These are the reactions, and A, B, C, D are the molecules. Okay, so and the same thing, we have assigned given values to each of the molecules. That means B1 reaction, that's the reaction name. B2 reaction, like that. So this is the A molecule, B molecule, C molecule. So row into columns we have taken. So A molecule, which is not involved in V1 reaction, which is not involved in V2 reaction. Okay, like that. And at the end you can see, so it is involved in B1 reaction. Only in that reaction it is involved, no other reactions in that. So as you can see here, so A, it has involved in B1 reaction, so incoming one. This is a different one actually, this is a different package. Okay, just as an example I'm showing here. So it has -1, sorry, +1 in B1 reaction, and -1 in V2 reaction, like this. And B, it has +1 in V2 reaction and -1 in V3 reaction, so like this. So we used to calculate for each of the reactions. So if it is 0, it's not involved in any reaction, it's not involved in that particular reaction. B is not involved in V1 reaction, not involved in V2 reaction. Okay, and D is involved in V1 reaction and V2 reaction and V3 reaction. So everything is outgoing. So from D, it is V1 is outgoing, V2 is outgoing, and V3 is outgoing. So like that. And all other remaining reactions, it's not involved in, like that. Okay, so this is the way we was prepared the digital values, numeric values, numeric data we have prepared. And what we are doing is, we were taken all this data into MATLAB software. So currently I'm showing you a Linux version of the MATLAB, like a freeware of the MATLAB kind of software. This is similar to MATLAB only, this is called as FreeMat. So here we were taking all those data. Okay, so we have taken in the form of a matrix. We have taken comma separated, and each row are separated by the colon. Okay, so this is how it displays. So the x variable contained that matrix. So we for calculating the single value decomposition for that matrix. So, there is a function called SVD in MATLAB. So we were using that this way. Okay, so this will output three different variables. So we'll take that as u comma s comma v. Okay, so we will assign it stored in those variables, and so this is how it displays. So u contains this data, this kind of values, different large matrix it contains. So s contains this matrix and v contains this one. So we don't understand anything of it properly, so we were plotting this matrix as a diagram. So p and v is the one we are taking. So he has some algorithm, he has referred some algorithm. He needed this kind of data to be represented in this way. So yeah, this is the graph we would get. So the pathway what we have taken and produced this matrix is a TGF beta signaling pathway. It has in there are many research articles that has been published based on this TGF beta signaling pathway, that says there is one signaling molecule that needs to be nullified, so to prevent some sort of diseases that in that the TGF pathway, beta pathway is involved in. So that is the TROP6 molecule, and as you can see, this is one graph, one molecule, 10th molecule, that is minus, having a very least value -8, and there is one more. Okay, so like this it will display, and when we trace it back to that value, so 10th molecule is some I. It was having its name was some TROP6 or something. So that was exactly similar what we have studied in other research articles. So this is another another way of calculating or finding out the what we can say, finding out the target proteins in the signaling pathways. So whatever the data we have observed through wet lab experiments can be easily put out in this way without any major expenses. Okay, this is the approach. So what our tool needs to do is, whenever the user loads the software pathway and opens that plugin, okay, that needs to display all the data. Okay, for each of, even you can mention the reaction IDs instead of v1, v2, v3 like that, and you can mention the names of the molecules only. So whether that molecule involved in that particular reaction, like that only you can mention. You can display whatever I'm showing here. So each of the reactions, it needs to be displayed. So, and for each of the reactions, what is the each of the molecule is involved in that reaction or not, that needs to be displayed. And once the data is present, and we need to extract this kind of data, numerical data, out of that table and put it to MATLAB, or we need to integrate this MATLAB SVD function, MATLAB's SVD function into that plugin. So this is the function SVD, it's called as a single value decomposition, SVD. So I will share that one article that I've shown you. I will share this one with you people, so you will understand what our exact algorithm needs to be prepared in that tool. Okay, and once we have that data, that vector needs to be plotted. Okay, in this way, that vector needs to be displayed in that plugin only. There should be, if possible, there should be control to increase or decrease the manipulate that, move that plot. So in this way, whatever we have, we can tell the users, we can use an external software also, like plotting software also for users, that users needs to be downloaded and installed it clearly. If it is not installed, it needs to be displayed some error message, that all the basic things needs to be there. That we will do that. And yeah, that's all. This is the algorithm I was explaining you. So hope this video is helpful for you. So if you have any clarification doubts, if you have, please call me for clarifications. And thank you. That's all.